Next up is the stoichiometry of the whole process of synthesizing a palmitic acid, also known as palmitate. So in the whole process, or even before the process, to start the process, we converted 7 acetyl-CoAs into 7 malonyl-CoAs. And each time we used 1 ATP, so 7 ATPs are used for this process. Then we had 7 cycles of the 4 reactions: condensation, reduction, dehydration, and again the second reduction. And this process used 14 NADPH molecules, 1 acetyl-CoA and 7 malonyl-CoAs, which we produced here, are used in the process. Now an important energy consumption is the transport of acetyl-CoA that we saw in the beginning as well, that acetyl-CoA was transported into the cytoplasm, and the cost of this process is 2 ATPs. The cost of fatty acid synthesis is mainly 3 ATPs per 2 carbon units that we attach, except for the first acetyl-CoA, which only requires 2 ATPs, because there is no first reaction here. In this case, we used acetyl-CoA. So in short, the net ATP consumption for making a palmitic acid is 23 ATPs in total. 7 ATPs are used right here for the conversion of acetyl-CoAs into malonyl-CoAs, plus 16 more 16 more ATPs are used, basically because we transported 8 acetyl-CoAs from mitochondria to cytoplasm. Each one requires 2, so multiplied with 8, which is mentioned here, will give us 16 ATPs plus 7. So total we need these 23 ATPs that will be used for making one palmitic acid. Next up is what we call palmitate processing, or making changes to the palmitic acid that we make, because there are different types of fatty acids that are required in the body. This one palmitic acid has 16 carbons in it, and has got no double bond in it, which is represented here. 16 carbons, 0 means no double bond, so it's a saturated fatty acid. Now we can elongate this fatty acid, add up 2 more carbons to it, and convert it into this stearate or stearic acid, which will have 18 carbons and no double bonds. And we can further elongate it into longer saturated fatty acid, add up 2 carbons at a time. We have very long chain fatty acids as well in the brain or the central nervous system, especially, which have 24, 26 or even more carbons. So this is the elongation. Or another change that we can make to this palmitic acid is desaturation, that is adding up a double bond to it. So from palmitate, without increasing the carbons, we can convert it into palmitoleate or palmitolic acid, which has the same 16 carbons but has one double bond present between carbon 9 and 10. Again, we can do desaturation at this step, from a stearate into oleate or oleic acid. Again, it will have the same carbons, 18, like stearic acid here, but it will have one double bond, which will be again present between carbon 9 and carbon 10. So this is the processing of palmitic acid, you can say. Elongating systems are usually present in endoplasmic reticulum and the mitochondria. So this elongation, adding up the carbons to it, is usually done by the endoplasmic reticulum and mitochondria, and each elongation adds up 2 carbons, as I just mentioned as well. Whereas the desaturation is usually done by the help of this enzyme desaturase, which is the most common one, and it introduces the double bond between carbon 9 and carbon 10, as we saw in both of these reactions, that the double bond was added between carbon 9 and carbon 10. So this is done by the desaturase enzyme. Next up is the synthesis of big fats or big lipids. The two main varieties are the TAGs and the phospholipids, the two main types of fats. So how do we make these big lipids? It's discussed here. So one important thing to begin with is this molecule glycerol 3-phosphate, which is a precursor for the backbone of both these TAGs and phospholipids. Do remember, fat or TAGs mean the same thing. And how do we get this glycerol 3-phosphate? Sources of glycerol 3-phosphate. Most of it basically comes from glycolysis with the help of this enzyme dehydrogenase. Some of the glycerol 3-phosphate we can directly synthesize from glycerol with the help of glycerol kinase enzyme. So this is the minor pathway in the liver and kidneys. The major pathway is this one, glycolysis with the help of dehydrogenase enzyme. So for making these big lipids, that is phospholipids and TAGs, we begin with the glycerol 3-phosphate here. It is going to undergo a reaction through the transferase and is converted into phosphatidic acid. So both of these molecules, glycerol 3-phosphate and the phosphatidic acid, are very important. Phosphatidic acid is also considered to be a precursor of the TAGs and the phospholipids. Precursor is a molecule from which we basically make another molecule. So these are the precursors of phospholipids and the TAGs. And this phospholipid is made up of the glycerol 3-phosphate, plus we attach two fatty acids with it, which are done with the help of this enzyme transferase. So once we have the phosphatidic acid, again it's a multi-step process. We can convert it into the glycerophospholipids. On the right side we can see phosphatidic acid getting converted into the glycerophospholipids, which are the main lipids for the cell membranes, or it will undergo a couple of reactions on the left side as well. First converted into the diacylglycerol and then converted into the triacylglycerol or the TAGs. So these are the two most common types of the big lipids. Both of these can be produced with the help of this phosphatidic acid. So this one is about the biosynthesis of the membrane phospholipids, and the main thing to remember is where it happens. It happens in the endoplasmic reticulum and the Golgi complex. And the whole process begins with a head group being attached to the phosphatidic acid or to the TAGs, and elimination of two water molecules happen. The whole process is known as condensation. After these phospholipids have been synthesized, they will be transported from the endoplasmic reticulum to the membrane by some vesicles, where they will be used up in making these membranes. So the last part of these lectures is about the regulation of the processes that we saw. So fatty acid biosynthesis regulation, regulation of the TAGs synthesis and their breakdown cycle as well, plus the role of gluconeogenesis enzyme in the fat or the adipose tissues. Regarding the fatty acid synthesis, the most important thing is mentioned at the top, that it is regulated through the ACC enzyme or acyl-CoA carboxylase enzyme, which is the rate limiting step of the whole pathway. And this ACC enzyme, which we can see in the center, converts the acetyl-CoA into the malonyl-CoA molecule. This is the reaction it catalyzes, and it is the regulation is of two types: allosteric regulation as well as the hormonal regulation. For allosteric regulation, we must remember that the enzyme ACC is feedback inhibited by the product of the pathway, which is palmitoyl-CoA. So palmitoyl-CoA, when it has high quantities in the cell, it will stop the enzyme ACC, whereas the citrate molecule is the activator of this enzyme. So citrate, when it is in high quantities, that will activate ACC. This is the allosteric regulation. Now the hormonal regulation is mainly through the two enzymes, that is glucagon and epinephrine, and both of these hormones inhibit the enzyme. So glucagon and epinephrine stop this enzyme through hormonal regulation. They will inactivate it through phosphorylation, so attach a phosphate group to the enzyme and inactivate it basically. So this is a slide that we discussed in the last lecture as well, which is about the regulation of these two pathways through the two hormones, that is glucagon and the second one, insulin. We already know that high blood glucose levels causes secretion of insulin. Insulin causes the activation of ACC enzymes. So ACC, as we just saw, converts acetyl-CoA to malonyl-CoA, and this malonyl-CoA will be used up in the fatty acid synthesis. So insulin or high blood glucose is going to have a positive effect on fatty acid synthesis. It activates it. At the same time, this insulin molecule, when it produces too much of this malonyl-CoA, it is also going to stop this transporter, which was the carnitine acyl transporter. Therefore, fatty acids cannot go inside the mitochondria. Therefore, the second effect of this insulin will be to stop or inhibit the beta oxidation. So it has a negative effect on it through this CAT1 enzyme. Now looking at the glucagon molecule, when we have low blood glucose levels, that is when glucagon is released, its effect is opposite to insulin. It inactivates the ACC. Therefore, its effect will be to stop the fatty acid synthesis. So it has a negative effect here, and it has a positive effect on the beta oxidation by stopping the effect of this malonyl-CoA molecule. All is done through this enzyme ACC. So the last slide is about the hormonal regulation of the TAG synthesis, or the triacylglycerol's full form. And the two hormones involved are again the same, insulin and glucagon. And glucagon, you can say, teams up with epinephrine. When we have a high carbohydrate or high protein diet, this will produce high amounts of glucose and amino acids in the digestive system. Insulin will be secreted in this scenario, and that will convert these glucose and amino acids into the acetyl-CoAs, and this will result in conversion of acetyl-CoAs into fatty acids, and eventually synthesizing the TAG molecules. A high carbohydrate or high protein diet with the help of insulin secretion results in increased synthesis of these TAGs, because we have too much extra glucose, and that result in too much extra acetyl-CoAs. So we can store it in the form of these fats, that is TAGs. Whereas on the other hand, glucagon and epinephrine, which are the opposite hormones of insulin, which are released whenever we are not eating enough food, or the blood glucose levels are low, that will result in the TAG breakdown basically, or mobilization of TAGs. So synthesis will be stopped and breakdown of the TAGs will start under the influence of glucagon and epinephrine.